So I'm going to try to provide a brief overview of some of the work that we're doing, some of the work that I'm currently engaged in Cincinnati, in an effort to convince you that those three things are interrelated. And I think they, by studying neural networks, which is a relatively new neuroimaging analytic technique, we can better understand how omega-3 fatty acids may contribute to the pathophysiology of major neuropsychiatric disorders, and perhaps better understand how omega-3s may actually help treat and or prevent these disorders. So one of the clues to when we try to understand the etiology of psychiatric disorders is basically retrospective data suggesting that a lot of the psychiatric disorders, their initial onset occurs during the adolescent period. And this is important for a number of reasons, and using this information to help guide us forward is the fact that this period is also associated with major neuroanatomical changes in the prefrontal cortex, particularly. And we know that the prefrontal cortex plays a very important role in regulating limbic structures, including the amygdala, which we know also plays a role in emotional processing. And that's a very dynamic developmental maturational process that occurs during this period when psychiatric disorders are initiated. Emerging evidence suggests that, neuroimaging evidence suggests that the adolescent period is also associated with rather dynamic changes in the interrelationship between the prefrontal cortex and the amygdala and how they communicate. And this ultimately is associated with how reactive the amygdala is to emotional processes. And for example, even in children that are subsyndromal, they don't have anxiety disorders, but instead exhibit elevated anxiety to demographically similar children, are exhibiting elevated functional connectivity between the prefrontal cortex and the amygdala. And we know, based on meta-analyses, now that one of the important things here, I think, is that using neuroimaging to better understand how omega-3s interact with neuropsychiatric disorders. And one of the reasons I think that is going to be important is because we now, after 20 years, have developed a relative consensus as to what specific disorders are associated with from a neuroimaging perspective. But what has emerged from this 20 years of neuroimaging research is that the prefrontal cortex to the amygdala, there's hyperactivity, hyperconnectivity, resulting in amygdala hyperreactivity to various emotional processes. And this, to me, suggests that perhaps the maturation of this process that occurs during peri-adolescent development may represent a therapeutic target in which we can intervene, particularly in at-risk kids, to help put trajectories back on a normal course. But how do we do that? What are we going to intervene with? Well, you may have guessed, one of the interesting things that has also emerged in parallel with this research is the fact that all of the major neuropsychiatric illnesses, based on meta-analyses, exhibit lower blood levels of DHA, in some cases EPA, which I think is very important. Again, many of these disorders emerge during peri-adolescent development. So we have that association. And this is just one study that we published a few years back, demonstrating that the EPA plus DHA levels in the red blood cells of individuals with BP, bipolar disorder, are significantly lower than healthy controls, and those that blood levels in individuals that are at ultra-high risk for developing bipolar disorder are also significantly lower. And those at high risk, i.e. they have a family member, a biological parent with bipolar disorder, which increases their risk of developing bipolar disorder, is somewhere in between. And these levels across all these subjects are associated with both manic and depressive symptoms. So moving to the brain now, we know from our studies as well as other studies that during the periadolescent period, prefrontal cortex DHA levels also exhibit a linear increase, suggesting, again, DHA or deficiency may contribute to some of these abnormal neurophysiological processes. And importantly, we find repeatedly that red blood cell DHA levels are highly correlated with rat brain DHA levels. And so what mediates interconnectivity between brain regions as well as within brain regions are synapses, as you may have guessed. And there's now a plethora of very interesting preclinical data indicating that developmental omega-3 deficiency results in a deficiency in synaptic plasticity within a number of different regions, including the hippocampus, and a significant reduction in synaptic number. And we also know that DHA dose-dependently increases synaptic dendritic spine density in animals and increases resilience to things that reduce synaptic density, including inflammatory processes. Interestingly, also, there's some evidence suggesting that DHA insufficiency also reduces synaptic pruning during normal neurodevelopmental processes in the developing visual system. And this is relevant because we know, at least in the rat, as I suggested in the intro there, as also occurs in adolescent humans, this period is associated with very robust synaptic pruning in the connections between the prefrontal cortex and the amygdala. Therefore, synaptic pruning and DHA's effects on synaptic pruning may have a significant impact on these developmental trajectories. So this leads to the hypothesis that developmental omega-3 insufficiency disrupts cortical limbic network maturation, which, as I suggested, is an endophenotype associated with psychiatric illness. And so this is a very recent study that we haven't published yet, but it's very interesting to us because it supports the proposition that omega-3 insufficiency is associated with significant reductions. If you look at the first rat brain, you can see the difference between the control animal and the omega-3 deficient animal, and this is a perinatal deficiency paradigm. And importantly, postnatal fish oil supplementation to the deficient animals is sufficient to completely restore functional connectivity within the prefrontal cortex, as well as restore prefrontal DHA levels. We also have some behavioral data suggesting that the circuit, prefrontal cortex to amygdala, is also disrupted in animals that are deficient through perinatal development, specifically impaired fear extinction, which is obviously relevant to anxiety disorders and PTSD. And also using rat neuroimaging with a 7 Tesla, we demonstrate that the functional connectivity, this is resting state, is significantly reduced in animals that have undergone omega-3 deficient diets over periadolescent development, suggesting that we have some kind of target engagement here. Previous studies have also demonstrated in non-human primates that there's significant changes that occur between the prefrontal cortex and temporal cortical regions as a consequence of omega-3 deficiency. We've previously demonstrated in kids with high versus low omega-3 levels in their blood, significant differences in functional connectivity between prefrontal cortex and temporal cortex. And this is a paper that's relatively old now, but we demonstrated that at least at lower statistical thresholding, prefrontal cortex activation increases substantially in response to an attention task, but corresponding reductions in temporal cortex activation are certainly consistent with increased inhibitory feedback and cortical limbic connectivity. So these data suggest that indeed, or support the proposition at least, that omega-3 supplementation will increase functional connectivity in cortical limbic networks in youth with depression. And so this is a study that we just completed and has just been accepted for publication, where we had high-risk kids, that is, they have a biological parent with bipolar disorder, substantially increasing their risk of developing bipolar disorder themselves, or at least a mood disorder, but also depressive symptoms, which again, we know from other data significantly increases their probability of developing a bipolar disorder. So this is a very interesting cohort to work with, particularly when we're investigating potential preventative interventions. And although we didn't see robust differences between our placebo in terms of depressive symptoms, we did see significant changes in global functioning measures as well as clinical ratings of global symptom improvement, CGI, which may in fact be a more sensitive measure to the positive effects of fish oil on depressive symptoms. But we did find also that significant changes in the fish oil group in the prefrontal cortex to the temporal cortex functional connectivity, and this is during visualization of emotionally negative pictures. And simultaneously, we have also seen significant reductions between the amygdala connectivity with the temporal cortex, which is sort of a top-up reduction, which we view as potentially a positive thing. And this is using graph theory technique, showing, similar to what I've shown you in the rats earlier, that the subjects receiving fish oil supplementation show significant increases in prefrontal cortex regions, as you can see here, including connectivity with the amygdala. So that went much faster than I thought it would. So perhaps this as well. So neuropsychiatric disorders, so these are my conclusions. Psychiatric disorders are associated with omega-3 deficits and abnormalities in cortical limbic functional connectivity. Developmental omega-3 fatty acids is required for optimal synaptic maturation and plasticity in rats. Developmental omega-3 insufficiency alters cortical limbic functional connectivity in rats, non-human primates, and healthy children. Omega-3 supplementation increases cortical limbic functional connectivity in adolescents with depression. So this leads to, this is obviously not conclusive, but it gives us clues into sort of how omega-3s may contribute to, or maybe sort of more causal relationships between the deficiency that we see in kids with omega-3 deficits and neurological processes which we know have been implicated in psychiatric illness. And ultimately then restoring functional connectivity in, or certainly normal developmental trajectories in these particular networks may contribute to the therapeutic effects of supplementation. Questions?